If every enzyme in your body stopped for just 1 second, the chemistry of life wouldn't slow down, it would collapse. Inside cells, enzymes are not just helpers. They are the control system of metabolism, governing reaction speed, direction, and coordination. To understand all enzymes, you need to understand 3 core layers: structure, kinetics, and regulation. First, structure defines function. Most enzymes are proteins folded into precise 3-dimensional shapes stabilized by weak interactions. This folding creates the active site, a microenvironment where chemistry is optimized. Within this site, enzymes use multiple catalytic strategies simultaneously. Acid-base catalysis. Amino acids donate or accept protons. Covalent catalysis. Temporary bonds form between enzyme and substrate. Metal-ion catalysis. Metals stabilize charges or participate in electron transfer. This is why enzymes can accelerate reactions by factors of 10 to 10 superscript 1 compared to uncatalyzed rates. Second, kinetics explains how fast enzymes work. The foundational model is Michaelis-Menten kinetics. It describes how reaction velocity depends on substrate concentration. 2 critical parameters emerge. Vmax, the maximum rate when the enzyme is saturated. Km, the substrate concentration at half Vmax, reflecting affinity. These parameters are not just numbers, they define metabolic behavior. Low Km means high affinity, allowing enzymes to function even when substrate levels are low. Enzymes accelerate reactions by lowering activation energy, but importantly, they do not change the thermodynamic equilibrium. They only help the system reach equilibrium faster. Third, regulation integrates enzymes into living systems. Cells cannot allow reactions to run freely, so enzyme activity is tightly controlled. Allosteric regulation. Molecules bind outside the active site, changing enzyme shape and activity. This is central in pathways like glycolysis. Covalent modification. Enzymes are turned on or off via phosphorylation. Feedback inhibition. The final product of a pathway inhibits an early enzyme, preventing waste. A classic example is phosphofructokinase 1, which acts as a metabolic gatekeeper, responding to cellular energy levels. Enzymes also depend on helpers called cofactors and coenzymes. Some require metal ions like iron or zinc. Others depend on organic molecules such as NAD+, which shuttle electrons in redox reactions. However, enzyme function is fragile. Changes in temperature or pH can disrupt structure through protein denaturation, eliminating activity entirely. This sensitivity explains why biological systems operate within narrow conditions. Finally, enzymes are organized into networks. No enzyme works alone. In pathways like Krebs cycle, each enzyme passes its product to the next, forming a continuous flow of matter and energy. This organization allows cells to extract energy efficiently and build complex molecules with precision. So when you think about enzymes, don't think of isolated molecules. Think of a coordinated, dynamic system controlling every chemical transformation that defines life, from DNA replication to thought itself. If you want more dense, science-based explanations like this, keep following.